Hello guys, today we'll be talking about diphtheria and we'll be talking about diphtheria under the following headings: introduction, causative agents, pathogenesis, epidemiology, risk factors, diagnosis, prevention and control, and treatment. What is diphtheria? Diphtheria is a highly contagious acute bacterial infection that primarily affects the upper respiratory tract, characterized by formation of a pseudomembrane on tonsils, the pharynx, and the larynx. Diphtheria can also affect the skin, leading to cutaneous diphtheria. Despite the availability of effective vaccination against diphtheria in many countries, diphtheria still remains a public health problem in low and middle income countries where immunization coverage is low. Without appropriate treatment, diphtheria can lead to severe complications including myocarditis, peripheral neuropathy, acute renal failure, as well as airway obstruction, which can be highly fatal. Diphtheria has a case fatality rate of about 5 to 10%, and it is significantly higher in younger children as well as in elderly individuals. Diphtheria is characterized as a vaccine preventable disease, yet outbreaks still occur, especially in areas experiencing conflict, population displacement, vaccine hesitancy, and healthcare system breakdown and collapse. The World Health Organization (WHO) emphasizes diphtheria surveillance and immunization as key strategies in elimination of the infectious disease diphtheria. Let's talk about causative agent of diphtheria. The causative agent of diphtheria is the Corynebacterium diphtheria. Corynebacterium diphtheria. Corynebacterium diphtheria is the causative agent of diphtheria. Corynebacterium diphtheria is a gram-positive, non-spore forming, non-motile, pleomorphic bacilli. Gram-positive because it takes up the primary stain, which is crystal violet, when subjected to gram staining procedure, and it is non-motile because it doesn't move under the microscope. It is non-spore forming because the Corynebacterium diphtheria doesn't form spores, it doesn't sporulate, and it is pleomorphic because it exists in various forms. It has about 4 biotypes. The Corynebacterium diphtheria has a club shape and it is usually arranged in angular formations resembling Chinese letters. So you have something like this. Under the microscope, you can have it like this. You can have something like this. This is just like a prototype of how it looks like under the microscope, resembling Chinese letters. Corynebacterium diphtheriae has about 4 biotypes. These 4 biotypes include gravis. Gravis is associated with severe diphtheria infection. There is also mitis. Mitis is associated with a mild diphtheria infection. Intermedius. Intermedius has an intermediate virulence. And there is also Belfanti. Belfanti is the most rare and it is less commonly toxigenic when compared to these other biotypes. So these are the 4 biotypes of Corynebacterium diphtheriae. The Corynebacterium diphtheriae causes pathogenesis. It causes infection or it causes its disease through the production of a toxin known as the diphtheria exotoxin. The diphtheria exotoxin is the primary virulence factor that Corynebacterium diphtheriae uses to carry out its pathogenicity of causing disease. But it is not all strains of Corynebacterium diphtheriae that is capable of producing the diphtheria exotoxin. And the strains that don't produce this diphtheria exotoxin are not pathogenic. They are not capable of causing disease. And it is only strains that are infected by a bacteriophage through a process known as lysogenic conversion that is capable of producing diphtheria exotoxin. We'll talk more about the lysogenic conversion when we talk about the pathogenesis of diphtheria. Some other species of Corynebacterium can also cause diphtheria-like infections. We have the Corynebacterium ulcerans, Corynebacterium ulcerans, and the Corynebacterium pseudotuberculosis. Pseudotuberculosis. This species, Corynebacterium ulcerans and Corynebacterium pseudotuberculosis, also cause diphtheria-like infections as well. Let's talk about the pathogenesis of diphtheria. The pathogenesis of diphtheria, rather, the pathogenesis of diphtheria commences when the bacteria, Corynebacterium diphtheria, enters into the body and begins to cause infection. The bacteria, Corynebacterium diphtheriae, usually is usually transmitted via respiratory droplets through the coughing and sneezing of infected individuals. It is also transmitted through direct contact with cutaneous infected lesions from infected persons. The bacteria enters the nose and mouth and adheres to the mucosa epithelial cells of the tonsils, the pharynx, and the larynx. The bacteria, Corynebacterium diphtheriae, then multiplies within the surface of the mucosa epithelium. Corynebacterium diphtheria is non-invasive, and as such, it rarely spreads or extends deep into tissue, or it rarely spreads into the bloodstream as well. It only localizes within the site of infection and produces toxin. It is important to note that it is not all strains of Corynebacterium diphtheriae that is capable of producing toxin. It is only the strains of Corynebacterium diphtheriae that is that is infected by a bacteriophage known as betaphage that has the capability of producing toxin. So a betaphage, it is only strains of Corynebacterium diphtheriae that has been infected by a bacteriophage. A bacteriophage is a virus that infects a bacterium. So it is only strains of Corynebacterium diphtheriae that has been infected by a bacteriophage known as a betaphage that has the capability of producing the diphtheria exotoxin. And the Corynebacterium diphtheria is infected by a betaphage through a process known as lysogenic conversion. Lysogenic conversion. So lysogenic conversion is basically a process by which a bacteriophage confers and integrates its viral DNA into the genome of a bacterium, right? It is a process by which a bacteriophage, in this case betaphage, implements and confers its viral DNA into the bacterium genome. And when it does that, it transfers sections of its viral DNA into the bacterial genome. And the section, the particular gene that is capable of causing the production of diphtheria exotoxin, in which the betaphage transfers to the Corynebacterium diphtheria, is the tox gene. The tox gene. The tox gene is the gene in which the betaphage, which is the bacteriophage, confers on the Corynebacterium diphtheriae that makes it possible for the Corynebacterium diphtheriae to produce the diphtheria exotoxin. Once this toxin has been produced, the toxin is regulated. Toxin production in Corynebacterium diphtheriae is regulated. Corynebacterium diphtheriae produces a protein known as a protein known as the diphtheria Diphtheria, diphtheria toxin repressor. The diphtheria toxin repressor is a protein produced by Corynebacterium diphtheria to regulate production of toxin. In the presence of excess iron, that is in environments with iron, iron activates this diphtheria toxin repressor, and then the iron diphtheria toxin repressor complex inhibits and shuts down toxin production. However, in the throat, where this pathogenesis of diphtheria takes place, iron production or an iron availability is very low because certain proteins such as the transferrin sequestrates this iron, and as such iron availability is very poor, and so there's no iron present for to activate the diphtheria toxin repressor, and as such toxin production continues, and toxin production is massive, leading to the pathogenesis of diphtheria. So I talked about transferrin as the protein that sequestrates iron in the throat, limiting the iron from activating the diphtheria toxin repressor to shut down production of toxin. So these toxins when produced cause death of epithelial cells within the throat. And the death of these epithelial cells leads to the activation of the immune system. Immune system gets activated and triggers the immune response and triggers the inflammatory response rather. So toxins causes or kills epithelial cells within the throat, and the death of epithelial cells within the throat triggers the immune response to cause or activate inflammatory response. With the inflammatory response causes white blood cells to come to the site of the toxin, and then these white blood cells aggregate, and so we have aggregation of white blood cells, aggregation of red blood cells, even fibrin, as well as dead bacterial cells to form a gray to black leathery thick coating known as a pseudomembrane. A pseudomembrane. So dead white blood cells, red blood cells, dead epithelial cells, and fibrin coagulates to form a a coating, a gray or black coating, leathery thick gray or black coating within the throat known as a pseudomembrane. Pseudomembrane is one of the distinguishing feature of diphtheria. This pseudomembrane, once produced, attaches to the underlying tissue within the throat, and it extends from the throat to the larynx and even to the trachea, causing mechanical airway obstruction, which causes suffocation and has a strangling effect. And it is this pseudomembrane that gives diphtheria the nickname the strangling angel of children, because the pseudomembrane covers the trachea, causing mechanical airway obstruction. It is important to note that Corynebacterium diphtheria usually is non-invasive. It localizes within the site of infection, and it is only the toxin that spreads into the bloodstream systemically, causing further complications. The toxins principally spread to the heart, causing cardiac toxicity. It also spreads to neurons, causing peripheral neuropathy, and in the kidneys, it causes tubular necrosis, leading to temporal kidney failure. So that is basically about the pathogenesis of diphtheria.